Well, hello students and welcome to this first video for the course BCH1111 and the first main biochemistry video for 1118. So I hope that you have now had the opportunity to cover the first topic in this course, which is basic chemistry. So you should have thought about chemical bonds, about water, about acids and bases, about um, chemical groups, all those basic chemistry things. I hope that you've covered. If not, please make sure that you do, otherwise you will become confused in the things that we're going to be talking about from now onwards. So we're going to start off for your first topic in the biochemistry course is about amino acids and proteins. And we're going to be covering that over the next few lectures. So here is the plan for what we are supposed to be covering. So in this lecture, I'm going to be talking about amino acids, their structure, the way they're classified, structures and functions. Next time, we'll be talking about amino acid base properties of amino acids and peptide bond. Then we'll think about how proteins are formed with primary, secondary, tertiary and quaternary structures. We'll think about classification of proteins and the functions of proteins. And we'll finish by looking at oxygen binding proteins as an example of a very specific type of protein carrying out a specific function. So that's the plan for the next few lectures, and these of course will all be recorded. So I thought I'd start off by just defining again what we mean when we say that we're studying biochemistry. So this is a dictionary definition I found of the word biochemistry. So you'll notice that the word has two parts of it, bio, which will remind you of biology, and chemistry, which reminds you of chemistry. Dictionary definition is that biochemistry is the branch of science concerned with the chemical and physiochemical processes that occur within living organisms. So I think the important part of this definition is living organisms. What we're thinking about when we talk about biochemistry is not something that happens in a test tube, although biochemistry can be made to happen in a test tube, but it's what happens in a living organism. It's the chemistry of biological organisms, if you like. So my definition of biochemistry is that it's what makes a cell work. How does a cell function? What goes on inside a cell that makes it able to carry out its functions as a cell? So I think that most of you have already studied chemistry and biology. Please don't think that because you know about biology and chemistry, you already know about biochemistry. It's different. It's more specialized. It's also different from organic and inorganic chemistry. You'll need some knowledge from organic chemistry to understand biochemistry, but organic chemistry is not biochemistry. You'll also be studying a course in physiology, and physiology and biochemistry have quite a lot of overlaps, but they almost come at the same topic from a different angle. So for instance, I've told you that we're going to be looking in a few lectures' time at oxygen carrying proteins. In physiology, you will be looking at blood and the blood system. Knowledge that you will gain works together to help you to understand, but we won't be teaching you exactly the same things. Okay, so we're going to start this semester with structural biochemistry. We're going to be looking at the structures of biological molecules, and I've just put some pictures of examples on this page that I hope you're already familiar with. Here is a protein represented here. This is actually a representation of hemoglobin. You'll be seeing a lot more of hemoglobin. This you might recognize is DNA, a nucleic acid. So a different type of molecule to protein. Here is a phospholipid bilayer. So phospholipid molecules associated together to make a membrane structure. And here is a carbohydrate made up of sugar units, glucose units. So all these things have one thing in common. They are large structures made from small building blocks. So I'm going to be talking about building blocks a lot as we think about these biological molecules. And I want to start by giving an example from the world that we can see around us, an example of a brick. You've all seen a brick. You know what a brick looks like. It's a simple shape, a cuboid shape. Many bricks are made and they all look exactly the same. I think this man is showing how strong his bricks are by standing on one. So a brick is a simple shape. It's a simple object. Bricks are all the same shape and size, but we can use these simple bricks and build them together to make many different things. So here is a large stack of bricks that have been made all stacked up together, and here they've been stacked together to make a shape that is somehow again like a brick, if you like. Many small cuboids put together to make a large cuboid. This isn't a very useful building, but these same bricks can be taken and used to build other things. So here are some houses built from bricks. Here's one that's still being built from these same cuboid bricks, used to build a round house here. And here there are some cuboid houses, if you like, again built from the same bricks. Here's a more complicated structure. This is going to be a school that's being built. So the bricks are being used, but in between here we'll have something that isn't bricks, perhaps doors and windows. And here is a very complex building built from brick. I don't know if you can see, but there are many bricks here. The driveway here is covered with brick. The bricks are used to build each of these buildings, tall buildings and smaller buildings. Some of the things that are not brick added on to add to the complexity, but altogether a complex structure built from simple objects, the cuboid brick. So this is the idea that I want you to have in your mind as we're talking about complex biological molecules, simple things being put together to make something more complex. If you're wondering, this building is actually the British Embassy in Kampala. Okay, so if I move on. So macromolecules are built in a similar way. So here we see the example that we're going to be starting with. Amino acids are being joined together in a long chain to make a polypeptide chain. Okay, we'll explain those words soon. Here we see this chain being folded up in a number of different ways, folded again in more complex ways, and finally producing a complex protein. So 20 amino acids, which are simple molecules, joined together in combinations, folded up and shaped to make a complex structure. And that is the basis of building macromolecules. So each of the amino acids is a molecule, okay? Put lots of amino acids together and we make a complex macromolecule. Okay, so it's not just proteins that are built in this way from amino acids. We'll also be moving on to look at carbohydrates, and you'll find that complex polysaccharide carbohydrate structures are built from simple monosaccharides like glucose or fructose or lactose. You'll be learning about these in a few weeks' time. And again, in a similar way, we'll be learning about lipids. And for instance, in a biological membrane, many lipid molecules associate together to make the much larger structure of the phospholipid bilayer membrane. And nucleic acids, DNA and RNA, are built from the simple building blocks of nucleotides, again joined in long chains to make a complex structure which is actually able to carry information. So please have that idea at the back of your mind as we're talking about proteins and as we're talking about these other large biological molecules, these macromolecules. Think of the way that complex large things can be built from simple small things, and try and understand how the small simple things associate together to make the complex large molecules. Okay, so another important point about the molecules and macromolecules that we're going to be studying is that each of them has a biological function. And that biological function we can understand a lot about it by looking at the structure of the molecules that carry out that function. For instance, when we come to study nucleic acids, we'll learn about the double helix structure of DNA and how important it is that that's a double structure, two strands carrying information. In a few lectures' time, we'll learn about hemoglobin and learn about how the structure of this protein allows it to carry out its function of carrying hemoglobin, of carrying oxygen around the body. Okay, so a pause for questions, and I hope you have watched the introductory video where I explained how we can ask questions on the forum on the learning management system. So please do note down any questions you have at this point and use that method to ask those questions. Okay, so I'm going to go on now to talk about amino acids, which are the building blocks of protein. So again, just to remind us of this building block concept, here's a small boy with his building blocks. I'm not sure what he's building here, perhaps a house or some sort of building. But you can see that he's put together lots of small, simple shapes to make a complex structure. Okay, so this is the structure of an amino acid. And every single amino acid has the same structure. It's what makes an amino acid an amino acid. So we're going to start by looking at the parts of this molecule. So first of all, notice the carbon in the center, the central carbon atom. This is known as the alpha carbon, and all the other parts of the molecule are attached to this carbon. Remember that carbon has 4 bonds that it can make. So a central carbon atom, and to that central carbon there are attached 4 different groups. The first of these is NH3, or an amino group. Of course, an amino group that gives the molecule its name, amino acid. Okay, it has an amino group. At this side, there is a carboxyl group, COOH, or in this case, COO minus, because we're looking at the ionized state. Okay, so this is also the ionized state of the amino group. So a carboxyl group at this side. And here we have something that's labeled as R. That stands for a variable side chain, or R group. And I said that each amino acid is the same, but this is the bit that's different in each amino acid. Okay, so every amino acid has the amino group, the carboxyl group, a hydrogen atom here, but the R group is different on each one, and that's why we have different amino acids. So notice the hydrogen as well, I didn't label that particularly, but the 4th bond here is occupied by a hydrogen atom. So this is the basic structure of every amino acid, 4 groups arranged around a central carbon atom. So here is another representation of the same thing. This is just what's called a ball and stick representation, which helps us to see the size of the different groups. I hope that you remember that hydrogen is a small atom. Nitrogen and carbon and oxygen are somehow similar sizes. And the R group, well, it depends which R group it is. We'll see soon, but some are very small and some are very large. Okay, so here we see the 4 groups again arranged around the alpha carbon atom. Okay, and I want you to notice this double bond between the carbon and the oxygen here. Okay, so pause for any questions. Do note them down if you need to. So here is another different representation of an amino acid that you might see in books. And this is trying to show that the shape is not just a flat shape, as we're seeing it written on a piece of paper, but it's actually a 3-dimensional shape. It's got depth as well. And this representation is trying to show that 2 of these bonds from the alpha carbon are coming towards us, and they are these ones that are shown solid and with this sort of shaped triangle. And these 2 bonds are going backwards, going into the plane of the paper, if you like, and they're shown by these striped or dotted bond lines, triangles. Okay, so 2 groups coming out of the paper towards us and 2 going into the paper behind us. And this arrangement is a tetrahedral arrangement. So here is a tetrahedral arrangement. Here is a carbon atom with a tetrahedral arrangement of atoms around it. This is actually methane, which has a carbon atom with 4 hydrogens arranged around it. So I hope that you can see the 3-dimensional structure with this space-filling model. And here it is represented in a flat model, if you like, but again using the dotted bonds to show something that's going behind, and the solid bond to show something that's coming out towards us, okay? And then these two are in the plane of the paper, if you like. So this is the sort of arrangement that the bonds, the different groups form around the alpha carbon. So here is again trying to show this in 3 dimensions, the alpha carbon here, the hydrogen atom sticking upwards here, and the R group here is coming out towards us. So it's sticking out of the plane of the paper, and then these two are going back into the plane of the paper, if you like. Okay, so a tetrahedral arrangement. And that's the same diagram just turned around. And I hope that you can see, because it has 4 different things attached to it, the alpha carbon has 2 different ways that these different groups could be arranged around it. That is described as being asymmetric. So the bonds to the groups around the alpha carbon in an amino acid are asymmetric. Because of this asymmetry, there are 2 different possible arrangements of the groups around the alpha carbon. And these are known as the L form and the D form. And when they're drawn here, I hope that you can see that they are mirror images. So if you had a model of one of these and held it up in front of a mirror, it would look like the other one. Yes, they have mirror symmetry. So I hope that you remember that mirror symmetry, turning it round, doesn't make the 2 things the same. Okay, it's only a reflection that makes them the same. So the L form and the D form of an amino acid are quite different. So this property of amino acid is known as being chiral, chirality. The alpha carbon is a chiral carbon atom, chiral. Okay, so that's a new word for you to remember, a chiral carbon atom. This word chiral comes from a Greek word which means hand. And I think that this is quite helpful to remind us that if we look at our 2 hands, they also have the same sort of mirror symmetry. Your right hand and your left hand are like reflections of each other. And however you turn your right hand around, you can't make it just the same as your left hand. Okay? You can play with your hands and convince yourself that that is true. Your left hand and your right hand are not the same. They have mirror symmetry. So here is an example of one amino acid, alanine. In alanine, the R group is a methyl group, CH3. Okay, so alanine has a CH3 group attached here, as well as the carboxyl group that all amino acids have. So 2 different forms of alanine, L-alanine and D-alanine. And I hope that you can see that this representation here, these 2 bonds are somehow coming out of the paper towards us, and these 2 are somehow going behind. And the same here, forward and backwards. Okay, and here is a representation with the solid and striped lines again, forward, backward, forward and backward. And here is a representation that is just showing them as the flat shape. So L-alanine and D-alanine are chemically identical. They have exactly the same atoms in them. They would have the same chemical reactions if you were carrying out reactions in a test tube with them. However, in a biological system, L-alanine and D-alanine are different. They are biologically distinct. So a biological system which is used to dealing with L amino acids would not be able to deal with D amino acids. Okay? And this is because biological interactions often depend on the exact shape of a molecule. And these molecules do not have the same shape. They have the same chemical composition, but they don't have the same physical shape. So in a biological system, they behave differently. So an example that you might like to think about is if you have a pair of gloves, perhaps some of you have used medical gloves, you have one made to fit your right hand and one made to fit your left hand. If you try to put your right hand into the left hand glove, well, perhaps you can somehow make it stretch to fit, but it's not a good fit. It's not the right shape for fitting together with your hand. So that's the same sort of idea when we put the two different forms of amino acid into interacting with another biological molecule, for instance, an enzyme. Okay, so the important thing to remember from this is that biological systems usually only use the L form of amino acids, the left-handed form, if you like. It's given that word because of the way that light is twisted by a solution. You might want to read more about that. But so in most biological systems, in most proteins that we'll be talking about, only the L forms are used. There are some bacterial cell walls that use the D form, but mostly only the L form is used. So having told you about the two forms, you can now mostly forget about the D form of amino acids and only concentrate on remembering about the L form. Okay, and a pause for questions again. Okay, so I've said that each amino acid has the same 4 types of group around it, but that the R group on each amino acid is different. And there are 20 different common R groups that are part of 20 common amino acids. I'll explain exactly what I mean by common soon. But these are the 20 amino acids that we'll be concentrating on. They are the 20 amino acids that make up proteins mostly. So here are the names of all 20 amino acids written out, and I hope that you're going to become very familiar with these names. Each of the names is also abbreviated with a 3-letter code. And you'll see that for most of the amino acids, the 3 letters are just the first 3 letters of the name. So alanine, ala, glycine, gly, valine, val, and so on. Some are different because there could be confusions otherwise. So isoleucine is I-L-E, I from iso, L-E from leucine. Tryptophan, you'll see, is TRP rather than TRY. Aspartic acid, glutamic acid, asparagine is ASN rather than ASP, because you can see here that there could be a confusion with aspartic acid. So 3-letter abbreviations, mostly the first 3 letters of the name of the amino acid, but with a few exceptions. And then there is also a system of 1-letter abbreviations, and you'll see later on, especially when we do some more molecular biology, why this shortened form is very useful. And here the abbreviations become a little bit less obvious because many amino acids start with the same letter: alanine, arginine, asparagine, aspartic acid, for instance, all begin with A. So alanine has the letter A as its abbreviation, but aspartic acid, which also begins with A, actually has D as its abbreviation. And I make it a bit easier to remember by trying to think aspartic acid rather than aspartic. That just helps me to remember the D a little bit. You can do similar things for some of the others. So phenylalanine, the P has been used up for proline. So phenylalanine begins with PH, so it has a F sound. So the abbreviation is F. Okay. Tryptophan has the abbreviation W. Here they're sort of starting to run out of ideas, I think. So again, to help me remember this, I like to say twiptophan. Okay, twiptophan with a W. Arginine actually begins with A, but AR, so it has an R sound. So its abbreviation is R. Lysine is K for no good reason at all that I can see, except that K hasn't been used for anything else. Glutamine, I like to say cutamine. Okay, and tyrosine takes the Y from the second letter. Okay, so 1-letter abbreviations, which can be quite difficult to remember, but if you just think about them and start to memorize them, you'll start to get to know just what the abbreviations, the 1-letter abbreviations, which amino acids they're referring to. Okay, so do ask any questions that you have on that. So the 20 different side chains of the 20 common amino acids, each one is different, and the differences in these side chains are what gives each of the 20 amino acids its distinctive properties, the properties that make it that particular amino acid and not one of the others. However, some of these side chains are similar and give the amino acids similar properties. So acid or basic amino acids, for instance. And these similarities mean that the amino acids are often classified into different groups. But there is a disagreement, even amongst textbooks that we use, exactly how to classify different amino acids. So here is a table from Leninger. If you go to the Leninger textbook, you will see this table. Here is just the 20 amino acids listed and some of their properties, which you will start to find useful as we go on to look at them more. So first thing to notice is that the 3-letter and 1-letter abbreviations are also on this table. But the properties that this table uses to group them are non-polar aliphatic R groups, which is glycine, alanine, proline, and so on. Aromatic R groups, 3 amino acids with an aromatic ring. Polar uncharged R groups, positively charged R groups, and negatively charged R groups. Okay, so that is a very common sort of chemical classification that is often used for amino acids. However, those chemical classifications are not the only ones, and sometimes in biological systems, the size of a side group can be more important than its actual chemical properties. So the side chain can be large or small or tiny, very, very small. It can be polar or non-polar. It can be aliphatic, which means that it has a carbon chain, or aromatic, which means that it has a carbon ring. It can be positively charged or negatively charged. It can be acid or neutral or basic. It can be hydrophilic or hydrophobic, or it might have something special about it in a number of different combinations. So to make things even more complicated, some of these properties can be thought of as distinct. So an amino acid either contains sulfur or does not contain sulfur. There are only 2 groups possible there. But other properties are continuous. So tiny, small, large. It's a continuous spectrum. Some properties depend on the environment of the amino acid, exactly where it is, whether it's free in solution or whether it's part of an amino acid chain in a protein. So the charge, for instance, can vary with the pH of the solution that it's in. And some properties change when the amino acid is in a protein. So, for instance, we'll learn about cysteine and cysteine. Okay, so another way of looking at the classifying amino acids is to think of it as a Venn diagram, like this. So here are different groupings. So here, for instance, is a category of small amino acids, and within that category is a category of tiny amino acids. Here is a category of hydrophobic amino acids, and here is a category of polar amino acids. So here, for instance, you can see that T-threonine is small and hydrophobic and polar. Okay, so that shows 3 different properties. Whereas here, glutamine is within the polar grouping, but not within the small or the hydrophobic grouping. Okay, so you might want to study that diagram and understand how the different properties of the R groups can work together to give each amino acid a different characteristic. So in biochemistry, amino acids tend to be classified according to the chemical properties of the side chain. That's what we saw from the table in Leninger. Whereas if we're thinking in terms of molecular biology, what is more important is how a particular amino acid behaves when it's joined into a protein. So as I talk about protein structure, I will be talking a lot about hydrophilic and hydrophobic amino acids, for instance, because they are very important in determining just how proteins fold up and take their shape. If you're thinking in medical terms, an important distinction is whether an amino acid is essential or non-essential. So essential amino acids cannot be made by the human body, whereas non-essential human beings can be made. So in other words, it is essential to have some amino acids as part of our diet because we can't make them. We have no other way of getting them otherwise, whereas others we can make ourselves. And some amino acids can even be useful as drugs. And I'll be mentioning some of those. An example I've given here is cysteine, which can be used to treat paracetamol overdoses. Okay, so here is a table from Leninger of essential and non-essential amino acids. You'll be learning more about this in BCH1111 in your second year for those who are going on to do that course. But you can see here from this table that the amino acids that we can easily make ourselves, non-essential, are alanine, asparagine, aspartate, glutamate, and serine, and then conditionally essential. These ones our bodies are able to make, but if the body system is challenged in some way, for instance, in young growing animals or humans, or during illness, we lose the ability to make enough of these amino acids. So these are the amino acids that need to be provided in the diet for sick people, but not for well people. And then these amino acids, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine, are essential. We need to have a source of these amino acids in our diet to give us enough to make the proteins that make up our bodies. Okay, so I'm just going to take a little bit of light relief now and play you the amino acid song. I'm having difficulty getting to the control here. Sorry. Okay, sorry. Now we can play it, I think. Glutamine and threonine and cysteine, tyrosine. These are players, serine too, as well as asparagine. Now we'll see which ones are charged electrically free. Sorry, I don't know why the video isn't playing properly. Okay, the amino acid song. Isoleucine, glycine, and there's phenylalanine. These are all non-covalent proteins as well. There's another letter where leucine ain't it swell. Glutamine and threonine and cysteine, tyrosine. These are players, serine too, as well as asparagine. Now we'll see which ones are charged electrically for you. Glutamic acid is a 1, aspartic acid 2. Arginine and histidine are basic and iron. As is lysine now done, the amino acids Amino acid song. Okay, so there you saw all of the structures and names of the amino acids in their different types. So here is the table we saw. This is actually from another textbook, Molecular Biology of the Cell, Albert's. So here are the names of the amino acids, 3 and 1 letter abbreviations, and then categorized into the types of their side chain. So negative charge, positive charge, uncharged polar. So these are classified as polar amino acids in this grouping, and these are all non-polar amino acids. So let's look at those again. Okay, so here's the words of the song, and I think I'll just play it again and show you where each of the amino acids is in this grouping. These are all as well. There's another land, seen ain't it swell. Glutamine and threonine and cysteine tyrosine. These are polar serine too, as well as ferrigine. Now we'll see which one's in charge electrically for you. Glutamic acid is 1, aspartic acid 2. Arginine and histidine are basic environment. As is lysine now done, amino acid song. Amino acid song. Okay, so as you can see, that song can help you to remember all the names and the groupings if you're the sort of person that can learn well from learning a song. So if you're that sort of person, please do. You might have also noticed there's a bit of disagreement about exactly where cysteine ought to go in the lists. Cysteine is an amino acid that's usually found in protein associated with 2 of them bonded together to make cysteine. Okay, so cysteine by itself has one set of properties, cysteine has another set of properties, and that's why there is somehow some disagreement about just how it's classified. We'll move on. Okay, so this is back to our groupings in Lehninger: non-polar aliphatic, aromatic, polar uncharged, positively and negatively charged R groups. So here you'll see that cysteine is put in the polar uncharged R groups, but with a note which says cysteine is generally classified as polar. That means Lehninger classifies it that way, despite having a positive index because of the self-hydryl group. So cysteine is a bit of an odd one. People put it in different categories, so don't worry about which textbook you read. Okay, another pause for questions. So the next thing I'm going to do is just go through each of the amino acids quite quickly. And I'm going to be telling you some things about each amino acid, but I also made out a handout with the 20 common amino acids arranged on the handout with a picture of their structure, of their R group, their name and their 3 and 1 letter abbreviation, a description of the type of chemical group that their R group is, and then just some notes of interest, perhaps, if there's anything that's a bit unusual about that amino acid. I've included it on this handout. So you can download that from the LMS. And if you're the sort of person who likes learning things by heart, learning amino acid structures and properties is one of the things that might be useful for you as you go through your biochemistry, if you know those names and structures and properties. So you can learn those if you want to, and that handout will be useful to you for that. Okay, so these are the 20 common amino acids. Please do also remember that there are also many uncommon amino acids. So, first one to talk about is glycine. This is the smallest amino acid, and its R group, shown in pink here, is a hydrogen atom, just a hydrogen atom by itself. So the amino acid itself is as small as it can be. It's known as a tiny amino acid. And actually, I've told you that amino acids have a chiral carbon, but because 2 of the groups on this amino acid are the same, it's the only one that does not have a chiral carbon. Okay, there is not an L and D form of glycine. Alanine, also tiny. It has a methyl group, CH3, as its R group. We've already looked a little bit at alanine. Proline has an unusual structure where the methyl group and the amino group, sorry, the carboxyl group and the amino group of the R group here are joined together into a ring, which actually officially makes it an amino acid. But the important thing in biology about this is that it means that this ring structure means that the whole molecule is less flexible, less bendy than the other amino acids. So it doesn't bend into shapes as easily. In proteins, it's sometimes called a helix breaker. When we come on to look at secondary structure in proteins, you'll be able to see why proline is important as the helix breaker. Valine, another quite small amino acid. I couldn't find anything very interesting about that one apart from the fact it's an essential amino acid. Leucine and isoleucine. So you can tell by the name, isoleucine is an isomer of leucine. So if you compare these two, you can see CH2, CH3, CH3. And isoleucine has the same groups, but in a different arrangement. They are isomers. So remember, isoleucine is an isomer of leucine. Methionine is one of the 2 amino acids that contain sulfur. And here the sulfur is in the middle of a chain. So it cannot easily react with another sulfur. But because it contains sulfur, it can be used in medicine to treat paracetamol overdoses. So this side group is quite large, so this is one of the large or bulky amino acids. Phenylalanine has a ring structure, an aromatic ring structure. And this ring structure means that it's one of the amino acids that absorbs ultraviolet light. So that can be useful in the laboratory for measuring protein concentrations, and it can also be useful biologically because absorbing ultraviolet light can then help it to be used as an energy source in chemical interactions, chemical reactions. In medicine, if there's a defect in phenylalanine metabolism, excess phenylalanine and its products can build up in the body in a condition called phenylketonuria, and this can have the effect of disturbing the body chemistry, especially brain chemistry, and cause mental defects, mental problems in developing children. Tryptophan, or tryptophan, to remember the W, is another aromatic amino acid with a ring structure. So again, it absorbs ultraviolet light. This one is part of a biochemical pathway that produces serotonin. So if an individual doesn't have enough tryptophan, they can't make enough serotonin, and it can affect their mental state. It can cause depression. Tyrosine, the third aromatic amino acid. So again, a ring, again absorbs ultraviolet light. This one can act as a precursor of melanin and adrenaline, so important hormones in the body. Serine, small r group, a hydroxyl group, so a tiny amino acid. Threonine, an essential amino acid. Cysteine, a very important amino acid in forming structures in proteins. So this is the second amino acid that contains sulfur, and you'll see that the sulfur here is at the end with just a bond to a hydrogen. So usually when a cysteine is found in a protein, it's bonded to a second cysteine to make a structure called a cysteine dimer. So two cysteines together, joined together by their sulfurs, a covalent bond between the two sulfur atoms is called cysteine, and that's the form that's usually found in proteins, and it can form a fixed tie together between two parts of a protein, as we'll see. Medically, the sulfur in it makes it useful in treating paracetamol overdoses and important in forming protein structure. Asparagine, glutamine, lysine. This is one of the basic amino acids, one of the amino acids with a positive charge. So it can be very important in interacting with other things that carry a negative charge. For instance, positively charged lysines are very important for interacting with negatively charged DNA. Arginine is a metabolite in the urea cycle, and if that isn't working properly, ammonia can build up and cause problems in babies especially. Histidine is often found as acting as a proton donor or protein acceptor at the active site of enzymes. So very important in the correct functioning of enzyme molecules. Aspartic acid, a hydrophilic amino acid. Glutamic acid, this is another of the negatively charged amino acids, and this one is hydrophilic as well, so often found on the outside of proteins. So that was just a very quick run through. I don't expect you to remember each of those sets of properties from what I've said, but do get yourself this handout and study it and think about the different properties of each of these amino acids. Okay, a pause for questions again. So just to look at these amino acid structures in their groupings, again, these are diagrams taken from Leninger textbook, so you can look at these for yourself. This is the grouping of non-polar aliphatic R groups. So glycine, alanine, proline, valine, leucine, isoleucine, and methionine. And there's quite a useful mnemonic to remember this group, VIP glam. So I have a picture in my mind of perhaps a glamorous film star, very important person stepping out onto the red carpet, VIP glam. So valine, isoleucine, proline, glycine, leucine, alanine, and methionine, non-polar aliphatic R groups. Here is the grouping of aromatic R groups, so phenylalanine, tyrosine, and tryptophan. I remember these because each of the names has a Y in it, although it's somehow complicated because there are some others that have a Y as well, but all these three have a Y in their name. Polar uncharged groups, serine, threonine, cysteine, asparagine, and glutamine. Positively charged R groups, lysine, arginine, and histidine. So these are the three, especially lysine and arginine, but are important in interacting with negatively charged DNA. And then the negatively charged R groups, aspartate or aspartic acid, and glutamate and glutamic acid. They are the same. Sometimes it's written as 8, sometimes as acid. Okay, so they are the two negatively charged acidic amino acids. So, any questions? So, the 20 common amino acids that we've just been talking about are called common because they are the ones that are found most commonly in proteins. And they're found most commonly in proteins because these are the 20 that are coded in DNA, in the DNA instructions to make the protein. These 20 amino acids have their presence in a protein coded into the DNA instructions. You'll learn exactly how that works next semester in BCH1111. Okay, so the 20 common amino acids are the ones that are actually written into proteins by the instructions in the DNA. However, there are also a lot of uncommon amino acids that are not specified by those DNA instructions. And I've got some examples here for you of different ones. So a lot of uncommon amino acids are made by a modification of a common one. And you can see here that each of these has one of the familiar names, proline, lysine, or glutamate as part of its name, but something else added to show that something in the molecule has been changed. So 4-hydroxyproline has a hydroxyl group added to carbon 4. 5-hydroxylysine has a hydroxyl group added to carbon 5. Okay, and so on. So post-translational modification of common amino acids to produce a variation in the amino acids found in proteins. Here is a couple of unusual ones. Desmosine is actually made of 4 lysines joined together to make a sort of ring structure in the middle. You'll see that each of these is a lysine, but they're joined together to make a bigger molecule, desmosine. Here is selenocysteine. This is the same as cysteine, except instead of having a sulfur atom here, it has a selenium atom. Okay, so quite unusual amino acid. An important change that can be made to amino acids are reversible modifications. So quite often groups are added or removed from amino acids to change them and to change their properties in the protein. And this can be important in controlling the function of that protein. So here, for instance, is a serine with a phosphate group added to it. This group can be added or removed very easily in a phosphorylation and dephosphorylation reaction. Again, methylation, methyl groups added or removed, or even large groups like an adenine group. This is a nucleic acid, a nucleotide being added or removed to a tyrosine. Okay, so these modifications are carried out by specific enzymes that add or take away these groups, and they are often involved in controlling the function of the proteins. Other uncommon amino acids are found as intermediates in biosynthetic pathways. So here are 2 examples, ornithine and citrulline. You might come across these as you look at some of the biochemical pathways. Altogether, there's over 300 different amino acids known, all taking this same structure of a central carbon and the tetrahedral shape, but differences on the R group. So please do remember that the 20 common amino acids are not the only ones, over 300 different ones known altogether. Any questions? Okay, so that is all that I wanted to cover in this lecture, but just to encourage you to look up more, to find out more. And amino acids have been known about for a long time, so there are good sections in the older textbooks that we have in the library as hard copy. So there are many copies of Leninger that look like this, or Stryer that look like this. And there is a good chapter 5 in Leninger, for instance, on amino acids that will give you all this information. If you have a soft copy textbook, it will be Leninger from either 2008, the 5th edition, or the 7th edition from 2017. And in this, chapter 3 is about amino acids, peptides, and proteins, and a link there that you might be able to use. Okay, so that's all for today. So I'm sorry that I had a few technical difficulties, but I hope you've been able to follow along with me.